Test shows gene editing tools work in dogs, but not efficiently. A pair of buff beagles may have the edge in doggy bodybuilding contests. Scientists in China changed the dog's genes to make the small hounds extra muscular. The dogs are the latest addition to a menagerie of animals, including pigs and monkeys, whose genes have been edited by scientists. The pup's genes were altered with a powerful technology called CRISPR-Cas9. Cas9 is an enzyme that cuts through DNA. CRISPRs are small pieces of RNA, a chemical cousin of DNA. The RNAs guide the Cas9 scissors to a specific spot on DNA. The enzyme then snips the DNA at that spot. Wherever Cas9 cuts the DNA, its host cell will try to repair the breach. It will either paste the cut ends together or copy unbroken DNA from another gene and then splice in this replacement piece. Tying together broken ends can result in mistakes that disable a gene. But in the dog study, those so-called mistakes were actually what the Chinese scientists had been aiming for. Linshu Lai works at the South China Institute for Stem Cell Biology and Regenerative Medicine in Guangzhou. His team decided to test whether CRISPR-Cas9 would work in dogs. These researchers used it to target the gene that makes myostatin. This myostatin protein normally keeps an animal's muscles from getting too big. Breaking the gene can cause muscles to bulk up. Natural mistakes in the gene, called mutations, work that way in Belgian blue cattle and dogs called bully whippets. These mutations have not caused those animals health problems. The researchers injected the new gene editing system into 35 beagle embryos. Of 27 puppies born, 2 had edited myostatin genes. The team reported its success October 12th in the Journal of Molecular Cell Biology. Most cells in an animal have 2 sets of chromosomes, and thus 2 sets of genes. One set comes from mom, the other is inherited from dad. These chromosomes provide all of an individual's DNA. Sometimes the copy of a gene from each chromosome set match one another. Other times they do not. One of the 2 dogs that had mutations in the myostatin gene was a female puppy named Tainzhou. She was named after a heaven dog that appears in Chinese myth. Both copies of the myostatin gene in all of her cells contained the edit. At 4 months, Tainzhou had more muscular thighs than an unedited sister. The second puppy carrying the new edit was male. He carries double mutations in most of his cells, but not all. He was named Hercules, after an ancient Roman hero noted for his strength. Alas, Hercules the Beagle was not more muscular than other 4-month-old puppies. But both Hercules and Tainzhou have packed on more muscle as they've grown. Lai says their fur may now be concealing how ripped they are. That the researchers could produce 2 puppies with edited myostatin genes shows that the gene scissors work in dogs. But the small share of puppies with the gene edit also shows that the technique is not very efficient in these animals. Lai says the process just needs to be improved. Next, Lai and his colleagues hope to make mutations in beagles that mimic natural genetic changes that play a role in Parkinson's disease and in human hearing loss. That might help scientists who study those diseases develop new therapies. It also might be possible to use the gene scissors to create dogs with specific features. But Lai says the researchers have no plans to make designer pets.